All right. Okay. All right, so just in terms of where we're at right now, like I said, I'll talk about lab at the end of lecture. Okay, so I'll leave that out for now. So let's just focus on lecture. So on Tuesday, we had a quiz on chapter 5, right? Towards the end of class, yes? So we'll go over that first. Chapter 5 homework was due on Tuesday, okay? So I'll probably go over that a little bit too. I mean, they're kind of just two simple questions that'll probably take us 2 seconds to go over. So I'll come back to that. So if if you're in this week, right, we're here, basically we're covering chapter 6, okay? So I'll start, kind of, I guess I'll open up the PowerPoint for chapter 6 and going over some of the stuff for chapter 6. Moving forward, right, you have an assignment for chapter 6, which is due on Tuesday, okay? So that's the next assignment that's due, chapter 6. Remember, the recorded lecture is there. We should be listening to those recorded lectures, and then we always have the review sheet out for the next exam, right? So next exam, exam 2, you have a couple weeks until that, right? So I have it scheduled for the week of October 19th, just so you guys know. And that will be chapters 5, 6, 7, and 8. So it's a pretty sort of dense exam, okay? So again, make sure you have that exam 2 review sheet, which is here. And you're always taking notes on that, or at least have it next to you when you're taking notes when you're listening to that recorded lecture. Any questions, guys? So like I said, for next week, your assignment is the chapter 6 homework. We will, what do I have for a quiz? Oh yeah, I have a chapter 6 quiz. So we'll do a chapter 6 quiz next week, probably Thursday, okay? But I'll let you know about that. But so be ready for a quiz next week as well, all right? But the homework is due Tuesday. Any questions, guys? No? Any questions on Zoom? Any questions in here? Y'all okay with assignments and stuff like that? Yeah? Okay. All right, so let's start off by going over that chapter 5 quiz, and then we'll just look at those 2 homework questions for chapter 5 and just make sure we all understand that, and then we'll move on. Like I said, I'll talk about lab later. Okay. Oh, I skipped the quiz. Where is it? Here it is. Oh, why is it doing that? Oh, that's not what I wanted to do. There we go. All right. Is that the password? Yeah, okay. All right, guys, so let's go over this quiz. So blank proteins help the body recognize when it is being invaded by pathogens or a pathogen so that an immune response can occur. What's the answer for those 4 choices that are listed there? What's the answer to that one? Yeah, say louder. Cell recognition, okay? So remember, this is pertaining to the different types of membrane proteins, right? Remember I went through all those different types of membrane protein? Cell recognition proteins, what type of proteins are those? You guys remember there was like another type of protein that I talked about? What makes them, I guess, unique compared to some of the other proteins? Anyone on Zoom remember? So remember they stick, sort of something sticks out towards the outside of the cell. Moses? Say louder. No, well, that's another type, but they're glycoproteins. You guys remember that? So they have carbohydrate chains that stick off of the protein. So it's a protein with a carbohydrate chain attached. And that carbohydrate chain sticks to the outside of the cell, and it acts like an identifier, essentially. So that's what enables the body to recognize self, right? Its own cells versus some foreign material like a pathogen. Okay, does that make sense? All right, so that is the correct answer, cell recognition. Okay, water crosses the plasma membrane through what type of channel there? What's the answer to that one? Aquaporin, good. Any questions about any of these guys on Zoom? Okay. Blank requires ATP. So what type of transport there requires ATP? Active, that's easy, right? Whenever it says active transport, it means it requires energy, right? Remember, ATP is essentially the energy of the cell. So the answer is active transport. All right, all the following are components of the cell membrane, except which thing that's listed there? Testosterone, good. So so I guess, okay, so let me ask you this, I guess. What other type of steroid actually is part of the membrane? Cholesterol. And so what's the function of cholesterol in the membrane? Yeah, it helps with the fluidity of the membrane. All right, good. So not testosterone, but cholesterol is part of the membrane. Testosterone is also a steroid hormone, or a steroid. Cholesterol is not a hormone. Carbon dioxide and oxygen can readily diffuse across the plasma membrane. Is that true or false? True. They're gases, right? And remember I showed you that there's that one figure that shows you a whole bunch of oxygen in the lung, right? And then the blood vessel next to it. And that the movement of oxygen and movement of carbon dioxide, it just moves by basic diffusion, right? And they can actually just diffuse across the plasma membrane. All right, good. So the answer here is true. In a blank solution, there is a higher concentration of solute outside of the cell compared to inside of the cell. So what solution is this? Say louder. Hypertonic, right? So hypertonic means there's a higher concentration of solute, okay, outside that cell. All right, and so if a red blood cell is in a hypertonic solution, what happens to it? Red blood cell in a hypertonic solution, what will happen? What is it? So if we have our red blood cell, right, and it's sitting in a hypertonic solution, we'll just use sodium ions on the outside to represent the solute, right? And then inside the cell, there's only a little bit of sodium, right? But there's more water, right? There's less water outside. Say louder. So think about the water first. So look at the concentration of water, right? There's more water inside than outside. So if water is going to move along its concentration gradient, right, from high to low, which way will water go? To the outside, right? There's more water here and less here, so it's going to move out. So if water moves out of the cell, what happens? It'll shrink. Yeah. Now, if it was the opposite, right, where you have a hypotonic solution, right, and so now you have more water outside, okay, and anyway, you got less water inside, right, and more water outside, right? So we say hypotonic, meaning there's less solute, right, less sodium ions here, but there's more water on the outside, right? In the inside, there's more solute, less water. So if we talk about the movement of water now, which way is water going to move? In, right? And if water moves into the cell, what happens? It can swell up and it can ultimately burst, okay? So red blood cells like to be in what type of solution? Neither of these. What type of solution are red blood cells most happy where you don't have any net movement of water? So in these two situations, it's going to be a problem with red blood cells, right? If too much water moves out or too much moves in. but the red blood cells want to be in a situation where the surrounding environment, right, is the same as the intracellular environment. And what do we call that? Isotonic, right? So then that means there is no net movement of water, and those red blood cells are perfectly happy. Okay, does that make sense, guys? Yeah, so make sure you understand that. So, so you guys on Zoom, I just drew on the board. Unfortunately, the camera doesn't go that way. An example of what happens to red blood cells in a hypertonic solution versus what happens to red blood cells when they're in a hypotonic solution. If you have no idea what I'm talking about, go back to that recorded lecture. Same thing here. If it was still a little confusing, just go back to the recorded lecture and listen to that part again, right? I also can pull up the lecture and explain that a little bit more after the quiz if somebody wants me to do that, all right? But make sure we understand that. Red blood cells want to be in an isotonic solution, which means that the outside, the extracellular environment is similar to the inside, and there's no net movement of water, okay? All right, so in a hypertonic solution, the water will be drawn out of the cell and the cell can shrivel up, okay? Not good for the red blood cell. And like in our lab next week, we're gonna actually have some sheep blood and kind of demonstrate those three situations: isotonic, hypertonic, and hypotonic. All right, if we look at this next question here, guys, blank is the diffusion of water across a selectively permeable membrane from high concentration to low concentration. So diffusion of water is what? Osmosis. Right, and so that's why when the example I just gave, we're thinking about the movement of water, okay? All right. So what about this question here? What type of proteins have a shape that allows only specific molecules to bind to them? And then once it binds, it leads to a cellular response. What's this answer here? Which one for this one?
Receptor.
Receptor? Yes. Receptor. All right, all the following are membrane proteins except which one? Listed there. What did you guys put for this one? Translation proteins. I mean, I just kind of. So carrier proteins, channel proteins, receptor proteins, those are all membrane proteins, right? And remember, there's a whole, part of chapter 5 that talks about those different types of membrane proteins. All right, during blank, a vesicle inside the cell fuses with the plasma membrane and releases its contents. So what is this, endocytosis or exocytosis? Exo, right? Release, exit, it's releasing its contents to the outside. All right. Okay, so we got 100, so that's good. So hopefully everyone did well on that quiz. Again, if not, that means you need to go back and review that material more, okay? All right, let's just look at the chapter 5 homework questions. There's only 2. They're pretty straightforward, I think. Okay, explain the role of proteins in the fluid mosaic model of the plasma membrane. So I think I usually get kind of multiple answers for this. So somebody tell me how you answered this question in your homework. You could have kind of gave like a more general answer or a more specific answer, I guess, right? Either is fine. So somebody tell me how you answered this. Go ahead, Brad.
I said proteins can be channel proteins which allow the passage of the molecules in the cell into the channel, or they can be carrier proteins that bind substances and exist with the passage of molecules.
Okay. All right. So you gave some specific examples of proteins. And, there's other membrane proteins too, right? Remember that long list of membrane proteins, all of those sit in the membrane, but that's fine. So if we just gave me a couple examples, that works. Somebody else, did somebody else put something different? So Brad said he just gave an example of channel proteins and carrier proteins. So somebody on Zoom, what did you put for this? Oh, we have no volume on this computer. Oh, that's not true. Live. Nobody's talking. Anyone on Zoom want to answer? Tell me what you put for your number one question here. I know one of you guys did the homework. Anyone?
I put
All right. I'll take
explain
that
the
as
role of proteins in food.
anyone else? I mean, did somebody put anything about like peripheral proteins and integral proteins? Anything like that? How'd you answer it?
I listed the proteins. I listed the proteins and their functions.
Somebody else, Moses, how'd you answer it?
I don't think she can hear us.
Yeah, she can't hear us.
She can't hear us?
I don't think so.
Okay.
Because I was trying to answer the question, but okay.
Okay. All right. The different types of, okay. All right. So that's fine. So again, that's kind of a general way to ask the question or whatever, but so anything that makes sense in terms of proteins in the memory, right? You could have listed all those different types of memory proteins. You could have just described a couple, that's fine, but remember that we need to know the different types of memory proteins that exist and the basic function of them, okay, for the exam, okay? All right, so if we look at that next one, yeah, so it says explain the difference between passive and active transport across the plasma membrane and give an example of each. So how would you describe passive transport across the membrane? So now we're talking about just across the membrane. Passive transport, what's passive transport? I mean, go ahead. Okay, good. So molecules move from higher concentration to low concentration. They move along their concentration gradient. Yep. All right, what about between passive and active transport? There's another difference. Right, active requires energy, passive doesn't, okay? Now, what about active transport in terms of the way molecules move? Do they move from high to low? Good, they move the opposite, from low to high. So remember that molecules have the tendency to move from high to low. If you want them to move in the other direction, it requires an input of energy, right? So it says to give an example. What example did you guys give? Moses. What did you say? What do you mean? Okay, that works. All right. Osmosis, yeah, that's diffusion of water, right? Moving from high to low, that's passive, right? What about active transport? Did anyone give, what did you give an example of that? I think I just kind of gave you guys one example of that. Good, the sodium potassium complex. There's other examples as well. There's some other like transport of other molecules like glucose and stuff like that. They involve, they're a little more complex and technically active, but the sodium potassium pump is our example for now. We'll just go with that, okay? Because it moves sodium against its gradient and also moves potassium against its concentration gradient. So whenever you're moving it against the gradient or from low to high, it requires energy. All right, good. Okay, any questions about anything that we kind of just started to talk about in chapter 5? Or anything that you want me to go over a little bit more? Are we okay with the hypertonic, the isotonic, and the hypotonic solutions? I think that sometimes is a little confusing. I mean, I just went over it again on the board, but you guys feel okay with that right now? Yeah? Okay. Any questions from anyone on Zoom about anything from chapter 5? So we just went over the chapter 5 homework, and we just went over that chapter 5 quiz. So anything else you guys want me to go over? All right. So let me pull up chapter 6. Actually, let's look at, one second, I'm going to do one more thing. I'm going to pull up this exam 2 review sheet. So again, we've only completed so far chapter 5. Now we're on chapter 6. So it's to your benefit to just be completing that as you move along, right? And then submitting it right before the exam. You know, and like I said, if you're copying and pasting stuff from the internet or from your friend, then that's not going to help you on the exam. So that's on you. Not a good way to approach the assignment. Let's see, can it open? Maybe. All right. Okay. So let's see if we kind of hit most of these things on chapter 5. Or take a look at that review sheet, guys, and just see if there's something that you're like, oh, can you go over that a little bit more, maybe? So we definitely want to make sure we know the components of the plasma membrane, right? So what's the main component of the plasma membrane? What's the main component of the plasma membrane? What makes the bilayer, Brad? Phospholipids, right? Good. And then there are proteins that sit within, you know, in between those phospholipids, right? There's peripheral proteins, there's integral proteins. There are also glycolipids and glycoproteins, right? I think I have I don't have everything listed there. So like for components of the plasma membrane on your review sheet, you should be listing the components. Maybe you draw it, whatever's going to help you understand. Also, what else we're missing? One other thing, cholesterol, right, is also one of those components of the membrane too. Okay, so we want to make sure we know all of those components. And then, like I said, the function of all those membrane proteins, we should know that. And then I gave examples. Remember, I told you don't need to memorize really all those examples, but you know, I could ask you kind of like, well, what if one of these wasn't functioning? What impact would that have, or something like that, right? And so those examples help you kind of come up with something there. But like I said, you can come up with your own example if you want, right? So it's not necessarily about memorizing those examples, but knowing the function. All right, and so they're all listed there, the different types of membrane proteins. So again, you should be able to fill in the function of those there, right? And maybe add the examples onto your review sheet. All right, so what does it mean to be selectively permeable? Moses. Yeah, it allows only some molecules to get in, right? And some, you know, or to get out or whatever, right? But others can't, okay? And it's regulated. It's not just going to let everything in. The cell only takes in what it needs and then needs to release its waste products, okay? It's not just going to, you know, take in everything all at once or anything like that. It only can handle what it needs, essentially. All right, we talked about diffusion, I think a decent amount. What else? Yeah, I think we kind of talked about everything related kind of to diffusion there. Remember, we first started talking about, or in the lecture, it's set up by just talking about basic diffusion, right? Just movement of molecules along their concentration gradient. Then we thought about it more in the realm of biology with the membrane, okay? And things moving across the membrane. And that's the hypotonic, isotonic examples. What for facilitated transport? What do we mean by that? What's facilitated transport? Facilitated transport. How would you describe that? Brad?
It's molecules that need the assistance of some other protein to get across the membrane.
Yeah, so like another protein like a carrier protein, correct. Or a channel protein. And does it require energy? No, because in that case, it's still moving along its concentration gradient, but it just has to get through the membrane through a carrier or a channel, okay? But remember, facilitated transport, though it may sound like it requires energy, it does not, okay? It's only when something moves against its gradient that it requires energy. Okay. All right, so I guess the last part, we talked a little bit about, I don't think I talked very long about this, but some different types of structures that are pretty much kind of made up of proteins that join cells together, right? So that's what that is down there. So it says animal cell surface. So adhesion junctions, tight junctions, and gap junctions. Those are all different types of connections between cells. And basically, it's membrane proteins that sort of link those cells together, okay? What's the extracellular matrix? Have that written there. Again, I don't think I talk about it too much, but what do we mean by extracellular? Well, extracellular means what? Outside the cell. So what's the extracellular matrix, Fred?
It's just a bunch of proteins with polysaccharide fragments.
Yeah, so yeah, it's really a bunch of kind of proteins like filamentous kind of proteins that sort of provide structure on sort of the outside. So if you think about like tissues like bone, right, or cartilage, these connective tissues that are strong, they have these certain stronger proteins in the extracellular matrix, right? And then the cells sit inside one. So it just provides a sort of means of structure. And membrane proteins will attach to that extracellular matrix, okay? And so again, if you talk about cells joining together to form a tissue, they will also associate with that extracellular matrix. So the idea is they don't just like float around, you know, there is like structure and they interact with things on the outside of the cell, okay? And that's that extracellular matrix. So I guess it's kind of like, it's more of a solid, more solid kind of structure than you might think, I guess. All right, so I'm going to pull up the lecture on chapter 6. If you guys take a look here, this is obviously the review sheet for chapter 6. And I think I told you guys, this is really kind of the setup chapter for the next two, which are on photosynthesis and cellular respiration. So everyone's favorite chapter is always the photosynthesis chapter. It's also my favorite chapter. I'm kidding. Anyway, so chapter 6, like I said, we'll talk about enzymes a lot, right? We'll also talk about some laws of thermodynamics, different forms of energy. We'll talk about ATP and the ATP cycle. And then we end talking about oxidation reduction reactions. So maybe if you guys are in chemistry right now, you talk about some of these things. I don't know. Okay, but it really is set up for the next two chapters. Okay, so I'll ask you this, I guess, right now. So what about enzymes? What do you guys know about enzymes? Why are enzymes so important to biology, I guess? Brad?
They help lower the amount of energy needed for that reaction to occur.
Yeah, they help lower the amount of energy needed for reactions to occur. Yeah, this energy of activation, right? So what if like there's an enzyme that's not functioning within the cell, right? Or an enzyme, just one enzyme within your body that's not functioning. What do you think that means for like overall health or disease? Do you think it would have an impact if it's just one enzyme? It does, right? So the idea is that these enzymes are specific to these reactions that they're going to catalyze. If your cell is not able to, complete a certain reaction, it has really detrimental effects and can lead to disease, right? So very serious disease too, not just like, something that's not serious. And that's just one enzyme, okay? So enzymes are obviously super, super important to the function of the cell, but also to the function of your organs, to the function of your body, okay? And again, remember, they're specific. They only catalyze one reaction, okay? So let me pull up chapter 6 lecture and just talk a little bit more about this, but again, you gotta listen to that recorded lecture and spend time with this material, okay? Maybe in high school you can kind of get away with not really doing work. Like, I don't think I did any work in high school, right? But then if you're going to be a science major in college, you have to do some work. I think it took me until probably sophomore year to realize that, so it's all right. I give you guys a break. But you have to put in some time, right? And it's not a crazy amount, but a little bit. Okay, guys. So, and if you don't understand something, it probably just means maybe you got to spend a little more time with it. That's all, right? And so remember, if you don't understand something, ask me, ask the tutor. Don't just keep going and not understanding it and then take the exam. That's not going to end well, okay? So just remember that I'm here. You can ask me questions, okay? And that's why you also have the tutoring sessions. If you feel more comfortable with an individual tutor, you can also request an individual tutor. But most of the time, it's really just about spending that time that you need with studying, okay? And it may be more than you think. All right, so let's pull this up. Why do I keep passing it? I'm all the way in chapter 9 and 10. We're not there yet. Okay, so chapter 6. I'm only gonna spend, I'll spend probably like 10 minutes or so, maybe 15 minutes. And then, like I said, I'll go over the lab assignments and things like that. And then we'll be done for today. Because like I said, you gotta really kind of sit and take the time and listen to that recorded lecture. Okay, so when you guys look right there at that title slide, it says metabolism. So what do we mean? What do you mean by metabolism here? I mean, how would you define metabolism kind of within the realm of our class, I guess? What's metabolism? I think I might define it on the first slide.
Isn't metabolism like the way that our body breaks down something so that we
What do you
can
think?
get vitamins
What's
out
the
of
sort of definition of metabolism if we're talking about like within the cell? Moses.
it? Can you hear us, Professor?
Yeah, can you hear us?
Say louder. Okay.
Professor!
She can't hear us?
Yeah, I don't think she could hear us.
Oh my gosh.
I sent her a message, but she didn't respond.
Yeah, I sent her a message in the chat, but she didn't see it.
Yeah, I don't know.
Should we send her an email and just like let her know?
Yeah, because I've been trying to answer the
Do
question.
you guys know somebody that's in person that you guys can text?
Yeah, I got some. I'm gonna text somebody in person.
Yeah, do that.
Because she's not even gonna watch the emails, like see the emails or text.
Okay. There's other types of reactions as well, but pretty much those are kind of the general categories, right? All right. The first part of this, like I said, talks about energy and sort of the flow of energy, okay? So this is kind of just general information on energy. You may know this already, some of this already. So we should know the difference between kinetic energy and potential energy, right? Kinetic energy is mechanical energy or energy of motion. Potential is stored energy, right? So if you think about, it says chemical energy, right? So molecules,
Hear us. And
Zoom
will
fucking
store
sucks.
energy, right? And if you break them down, a lot of times it will release energy. And then the cell can use that energy, yes.
Somebody that's in her class.
if anyone has Moses' number,
Oh, I texted somebody in the class to let her know.
Yeah, I text
Oh.
Brian.
Got it.
Professor,
I cannot hear you.
Can you hear
She's
us?
checking it
Don't
now.
ask me
Can you
why.
hear us now?
Let me see something.
Are we
1 second, guys.
are
You're
muted?
muted.
I know why, maybe. Yep. Go ahead.
Hey.
Now I can-
Can you hear us now?
Can you hear us now?
Hear
Now
us?
I can hear. Yes.
Okay.
Okay.
Yeah. So when I asked you if you had questions, anyone got questions from like half an hour ago?
Yeah, when you were asking us the questions about chapter 5 homework, I was trying to answer it.
Okay. All right. I got you. But does anyone have any questions in terms of wanting me to clarify anything else? We're okay right now?
I do. The mini lab assignment is
I'm going to talk about lab when we're done with this, okay?
okay.
I'll 10 minutes or so. Other questions? All right. Okay, so I can hear you guys loud and clear now. So just be like, hey, I have a question or whatever, because once I walk over here, then I can't see you because of the lovely way that this is set up. All right, sorry about that. Thanks, Brad. Okay, I was wondering why nobody was saying anything. I didn't think you guys would do that. That disconnected. All right, so if we talk about the flow of energy, that's what this next slide shows. And again, I don't want to go through everything here because I want to just introduce some of the things towards the end. You know, this is kind of this general flow of energy that we're going to talk about some of these different processes in the next couple chapters, right? So you got solar energy there coming into the plant. So what does that represent? the solar energy coming into the plant there, I guess. And then you also see some stuff being dissipated off as heat, right? The idea that really energy is never lost, it just changes forms, right? And there's no chemical reactions that are completely 100% efficient, right? So you'll always sort of lose some stuff in the form of heat, right? With that solar energy going into the plant, what process does that represent there? Photosynthesis, right? And then it's basically showing, okay, well, so now this plant can make its own food, right? Its own carbohydrates that are stored, right? There's energy stored in those molecules. And when, I guess that's a moose, when that moose eats that plant, right? And digest that, then he can break that down to form mechanical energy that he can use for movement, okay? So that's just kind of a really sort of general kind of picture to show a little bit of flow of energy, right? All right, so the next couple of slides talk about some laws of thermodynamics, okay? So I just kind of mentioned this, right? Energy is never lost, never destroyed, it just changes forms, okay? And one of those changing forms can be heat. I already showed you, I already said this, okay? So now this is basically illustrating what I just said, okay? Okay? The second law of thermodynamics has to do with disorganization. Now, I'm not gonna go through this right now. Listen to the recorded lecture of this part, just because I feel like it takes a little bit of time to explain it properly. So that I don't want to just explain it like halfway and then everyone be confused. So in the recorded lecture, I explain it, make sure you guys listen to that and really be able to understand what these figures are showing here, okay? So it's about this idea of entropy, okay, or disorganization, okay? And then that has to do with stability, right? And kind of dictates how those molecules will sort of behave or what they have the tendency to do, okay? So like I said, make sure you guys spend enough time looking at this and listening to this part, okay? All right, now, what I wanna at least just talk about a little bit more is, let's see, what do we wanna talk about? I guess we can mention this. All right. So if we think about metabolic reactions in the cell, right? We think about reactions, a lot of them require energy, right? Some of them don't require energy, right? Some of them will give off energy. And what was my example that I gave in general? If you think about degradation reactions or synthesis reactions, like we talked about in chapter 3, I think that was, right? Which ones do you think will give off energy? Right. Degradation, right? If you're going to break something down, and I just told you that molecules have stored energy, right? And we just said the moose broke down those carbohydrates to get energy. So in general, if you're breaking down something or degrading something, it's going to liberate energy, okay? And what do we call that? What type of reaction? Here, that's listed. Let's move my little... So what type of reaction? One of you guys on Zoom, you can answer that. Which type of reaction, exergonic or endergonic, releases energy, you think?
Exergonic.
Yeah, right?
Exergonic.
Okay. All right, thank you. So exergonic, right, products have less free energy than the reactants, okay? So you're ending up with more energy than you started with, right? Endergonic reactions will... Sorry, what'd you say?
What was the answer to that? You said exergonic?
Exergonic, yeah.
Thank you.
Yep. Endergonic reactions will require input of energy. So those types of, those are synthesis, right? If you're going to build something, do you guys agree that it requires energy to be put in? Okay? Same idea. All right. So that free energy that's listed there, we call that delta G. It's not chemistry, so I kind of try to keep, as much chemistry sort of out of it, but obviously we have to talk about reactions and chemical reactions inside the cell. But really what I want you guys to know is that exergonic reactions give off energy and endergonic reactions require the input of energy. Okay? As simple as that. All right. Now, if we're going to talk about metabolism in the cell, you have to talk about ATP, right? Because remember, ATP is the energy of the cell. So how do you think the cell, so if a reaction requires energy, right, where does the cell get that energy from? Not really a trick question. It gets it from ATP, right? How? It essentially couples reactions that need energy with the breakdown of ATP, right? So we said if you degrade something, you release energy. So if you break down ATP, which I'm going to show you, it liberates energy. So if you sort of pair that with the reaction that needs energy, that's how the cell is able to drive those reactions. Okay, does that make sense? So when we say ATP is the energy of the cell, this is giving you a little more detail about that, okay? So that's what this is saying about coupled reactions. So if you guys look here, this is this ATP cycle. You guys should understand this. We're starting off there with what? ATP. What does ATP stand for? Adenosine triphosphate. So why is it called triphosphate? It's got 3 phosphates there, right? And so now if we continue along here, right? And we're gonna break down ATP. We break it down by doing what? Cleaving off that last phosphate, right? And you end up with, oh, that's a typo. It's supposed to be ADP plus a phosphate. Oh, I never noticed that before, I don't think. Let me see something. Oh, now it switched to ADP. Okay, sorry. I don't know why I did that. All right, so anyway, right, ADP plus a phosphate, adenosine diphosphate, right, and then a free phosphate, because you cleave that off. Once you cleave that bond and release that one phosphate, it gives off energy. It's an exergonic reaction. You guys see that? Okay? So the cell can use the energy that's derived from this to drive other reactions, like for the synthesis of proteins or something like that. Okay? Now, what would happen, I guess, in the cell if you were never able to convert that ADP back to ATP? Do you think that would be a problem? Yeah, I mean, the cell needs to be able to sort of regenerate that so that it has more energy available, right? So basically, it adds another phosphate, and then there's your ATP back again. That's going to be an endergonic reaction itself too, right? Because you're synthesizing something and adding that phosphate group. So when we say ATP cycle, this is what we're referring to. Starting with ATP, breaking it down to ADP, and then going back to ATP. Okay? Does anyone have questions about that? No? All right. Okay, so this is just giving you an example of essentially muscle contraction and how it requires ATP. Okay? All right. The last part of the lecture talks about enzymes or focuses on enzymes, and then there's a little bit on oxidation and reduction reactions, like I said. Right? Let's see here. Okay, so I guess this is worth talking about a little bit. So this mentions something called the metabolic pathway, right? And this is the way the cell operates, okay? If, you know, A is the initial reactant, and what the cell is ultimately wanting to produce is G, okay? Each one of those arrows is another reaction, okay? Why doesn't the cell just have one reaction going from A to G? Why does it waste all its time with all those intermediate steps? What do you guys think about that? I mean, just think of, I don't know, come up with some reasons. You got right.
I don't know, it takes less energy to watch a little reaction to the super.
Okay, all right. Moses, were you going to say something? Okay, so that it needs to do those little reactions to even get to that point. Yeah, I mean, both of you are kind of correct. Also, a lot of times when you have metabolic pathways, like say C there, C might be a precursor for another pathway, right? And so there's often like branching off of these pathways. So that's part of it, but in other regulatory points where like it can basically, you know, you can inhibit this enzyme will stop the entire pathway, but maybe D is also a useful molecule, right? So that maybe this part runs, but then it can cut it off at this point, right? So it allows for a lot more regulation, right? And a lot more control of what's going on, okay? So good. Okay, so that's that when we say metabolic pathway, that's what we mean, okay? Each one of those arrows is a reaction that's catalyzed by a specific enzyme, okay? So now if you look here, you can see I added those enzymes in, okay? So can enzyme 1 catalyze the reaction going from F to G? You guys on Zoom can answer this too. Can enzyme 1 catalyze the reaction going from F to G?
No.
No. Enzyme 1 is specific for the reaction of going from A to B, okay? They don't substitute for one another, super specific. So if you are missing in your body for whatever reason, because of a mutation or something like that, there's nothing that can substitute for it, okay? Now, that's not always true. There are some examples where you have like a family of enzymes and they're only slightly different and maybe you can get compensation. But in general, we want to think of enzymes being super specific, okay? So if you're missing one of them, there's nothing you can do about it, okay? All right. Okay, the next part of it talks about enzyme substrate relationships and then talks about different factors that will impact enzyme activity, okay? And we're actually, so next week we're doing a lab on diffusion and osmosis, but the week after there's an enzyme lab. It's actually just gonna be virtual, it's not gonna be a live lab, but that will demonstrate some of these things that are outlined there, okay? In terms of factors that will affect enzyme speed, right, or enzyme activity, okay? And some of them are listed here. All right, guys, and so make sure, again, you spend some time with this in terms of understanding those factors. And then the last part of this, so there's definitely a lot more to talk about with enzymes, obviously. So I guess let me just quickly kind of say something about this because we were just talking about those metabolic pathways, right? So if you guys look at that metabolic pathway, okay, here, now you see there's kind of something else added in here, right? So see, F is your end product, right? So now the cell went through all those reactions and produced F. Maybe F is testosterone, right? Some hormone, okay? Now basically there's enough testosterone produced, right, in the body. So what does the body want to do? You want to stop that synthesis. You don't want to make more, right? Because you need to keep it at homeostatic levels, okay? Too much, not good. Too little is not good either. So the end product, right, or whatever synthesized, say in this case testosterone, it can actually turn off its own production, okay? So this is showing you non-competitive inhibition, okay? So like I said, in the recorded lecture, I talk about this and explain it in more detail, but basically, the end product, the testosterone, will bind to the enzyme, right? That's the first step in the pathway. And it will basically inactivate that enzyme, okay? So that no more of it is being made. All right, guys? So make sure you know, understand that. And again, you know, next week in class on Tuesday, I can go over that more if you guys feel like you need me to, okay? All right, like I said, the last part of this talks about oxidation and reduction reactions. Just a little intro to it. In chapter 7 and 8, we will talk about oxidation and reduction reactions again and again. And you probably won't want to hear anything else about oxidation and reduction after those 2 chapters, okay? For us, and again, if you talk about this in chemistry, you may really think about the loss or gain of electrons. For us, think about it as the loss of a hydrogen ion or the gain of a hydrogen ion. So you can actually look at a reaction and follow where the hydrogens go to determine what's oxidized and what's reduced, okay? All right, any questions right now about anything that I just sort of kind of introduced for chapter 6? All right, so again, I left out a lot of details, right? So like that doesn't count as like, you still need to listen to that recorded lecture because there's a lot of details that I skipped, okay guys? All right, so let me just talk about lab and lab assignments right now, and then that way if people have questions you can ask. So that, you know, so those of you guys that are in Thursday lab, I'm doing that now. So nobody has to come back at 2 o'clock or log into the lab Zoom or anything at 2 o'clock because we're gonna do it right now, okay? All right, so first things first, for you guys on Tuesday, I was telling you I wasn't sure what I was gonna do with the next lab, right? Because my link to the video that I had used for this virtual lab wasn't working. So anyway, it doesn't matter, okay? For next week, so for this week, essentially there is, you can see mine are hidden, right? If you guys look on Moodle for this week, for lab, there is nothing, okay? And that's accurate. For next week, okay, we're doing the in-person diffusion and osmosis lab. That's the only lab we're doing, okay? So that means you're either coming in to do it or you are on Zoom. If you are on Zoom, it is your responsibility to say, hey, Brad, can you explain that to me, right? Or hey, Moses, what did you do there, okay? I will make sure the camera is there. If you think it's better to FaceTime with somebody that's there to do it, that's fine. But it's your responsibility to do that. Not for nothing, but 90% of you said you were gonna come to lab, and unfortunately, it's probably 30% that have come, right? So whatever, that's fine, things might have changed, but you have to make sure you know what's going on, okay? So I'll just kind of make you aware of that. So for next week, the only thing that you need to do is basically have this lab protocol here. See this PDF, diffusion and osmosis lab protocol, ready. So if you're on Zoom, have it ready either, you know, on your computer and then you're taking notes or whatever. If you're live, I'll print it out for you. Okay, so I have printed copies for you guys if you're live. And then the assignment will be this mini lab report. So you won't be handing in that, using Cami to hand in that really large file, okay? You're gonna be following that as the protocol and writing down notes, but you're gonna do a mini lab report again, okay? So the assignment, the lab assignment for next week's lab will be this mini lab report, okay? And I'll explain that all when we're doing the lab next week, right? So the only thing you need to worry about this week is to have this lab protocol ready. If you're coming in person, I will have it printed for you, but it might be a good idea to look it over ahead of time, okay? So you know kind of what to expect. Does anyone have any questions about that for next week's lab? Moses. Yeah, you're gonna be doing it in lab, so you don't need to do anything ahead of time. Except if you want to look at it, you know, if you don't need to do anything ahead of time, you're gonna be following that protocol, and I'll tell you there's certain things that we're gonna everything in there. But I'll tell you what to do, everything will be set up, and then you'll just be collecting your data, right, everybody does your observations, and then you're gonna be putting that in the lab. Okay? You're gonna be after you do the lab. Yes, exactly. So you don't have to worry about anything next, okay? All right, so that's for next week. The other thing that I want to point out is I started grading things, okay? And I apologize, I'm a little behind, but there's a lot of you guys in this class. So, and I'll pretty much by tomorrow, I will have this metric and microscope lab done. Now remember, there was a virtual component, right, that we all were supposed to do before we came to the in-person lab, which is this, and you were submitting this assignment. Most people had to email that to me. I have it, don't worry, okay? If you emailed it to me, I have it. I'm going through and putting the grades in now. Some people were missing a couple of things from that lab or whatever, and if there was something missing, I made a note of it, and I gave you a grade, okay? I think there might have been 1 or 2 people that put the in-person lab there and then didn't hand anything in for the in-person lab. The in-person lab is different than this, okay? The videos that are here explain everything, okay? So listen or watch the video. It's not just listening, it's watching it. Most people did this assignment, okay? But I just want to make sure that it was clear. And then from last week, remember this chemical composition of cells lab, this mini lab report was, again, based on my demo, okay? Everyone should have already handed that in, okay? This is the in-person lab assignment from the metric and microscope. That's different. If you were in person, you just did it and handed it to me, and that's it, you got 100, okay? If you weren't, and the answers that you put didn't make sense, and that means you weren't paying attention when we were doing it, right? And so it was graded accordingly. So if you have a question about anything that I graded or my comments, please contact me individually, okay? So again, that mini lab report should have already been submitted for chemical composition of cells, right? And then again, moving forward, oh, and then also the cell structure and function, that was just labeling diagrams, okay? I will, before we have the lab exam, okay, so if you notice there's a lab exam, and I have it scheduled for the week of October 12th. We will review for this. I'll go over the lab assignments that you did as the review. We will review. There is also a review sheet that's there, okay? So I just wanted to make everyone sort of aware of kind of what was due and what. And so if you're still confused, let me know. I'll stay on Zoom. I'll stay here for a little while longer. But otherwise, you guys are all good to go, okay? So make sure, listen to chapter 6. Chapter 6 homework, be ready for a chapter 6 quiz next week, right? All right, guys. So like I said, if you have questions about lab stuff or about anything, I'm gonna stay on. Let me stop recording though.